All right. Alrighty.
Oh yeah. Okay, I see it.
Cool. Okay.
My sisters are here.
Right, still have a couple of minutes. Hi everyone, how are you? Hi Reginald, nice to see you again.
Hello.
Hey. All right. I'd like to try get a screenshot for audience. If you would like to turn on your video, that'll be great. Yeah, yeah. Hi, George.
Good morning, George.
Okay. Okay. All right. Could everyone turn on their video for a minute, just for a quick screenshot? Okay. Let's quickly do this. We still have time. In a minute, we can start. Alrighty. All right. All right. Thank you. And if you can now turn off your video, so we just less interference for the recording, that would be awesome. Okay. Just gonna do that. And I'll mute Kelly and Laura. Okay. Okay, exactly 8 a.m. That's great. Let's start our meeting. Yeah, oh, wait a second. Oh, that's the desktop. All right. All right. Hi, everyone. Welcome to the WebEV Talks series. So for you who's been here before, we have every Thursday and every Mondays in US or Tuesday in Australia. So today, the host for this talk is myself. I'm Carolina Sukmaji, as well as Elham Behesti, who's being a guest co-host with us today. So if you are interested to present in WebEV Talk, please email us, and you can find us in social media, and you can also direct message me through social media. So today is a great pleasure to introduce Kellie Machlus and Laura Goldberg. They're from Harvard Medical School and in US. Kellie's research is focused on how inflammation alters hematopoiesis and specifically its impact on megakaryocyte differentiation and platelet production. So her talk today will focus on a novel role for platelet-derived vesicles in bone marrow environment. Laura will talk about her preliminary data suggesting a possible role on, of bone marrow-derived extracellular vesicles in modifying murine hematopoietic stem cell aging phenotype. So without further ado, I hand it over to Kellie to start with her talk.
Great. Okay, hi everybody. Thank you for logging in, and thank you to Carolina for the invitation and for putting this together. I'm a bit new to the EV field, so it's really been nice to have this as a resource. And I actually run my own webinar series, so I know how much work it is. So it's nice to be able to kind of see people. All right, so this is quite a diverse crowd. So I am going to start with a little bit of background to make sure that we're all kind of on the same page. And it's going to be, this talk is going to be largely about platelets and platelet extracellular vesicles, or EV. So what are platelets? Platelets are a circulating blood cell, and you probably know them as the bandages of the blood. And the reason for that is they perform this function that you can see here, which is that they stick to vascular breach and they physically spread and aggregate together and really plug any holes. So not only for things like hemorrhage, but just maintaining normal vascular integrity, these small tears that happen all the time. So if you have really low platelet counts, you have thrombocytopenia and are at risk for hemorrhage. So platelets are anucleate and about 1 to 2 microns in diameter, and we have nearly 1 trillion platelets in circulation. Their half-life is only about 10 days, so we're constantly having platelets made and cleared from circulation. One of the things that's often talked about less is the other roles of platelets, and this is really one of the key discussion points of this talk and focuses of our lab. And part of that is due to their cargo. So what you can see here is a TEM image, and you can see all these granules in the platelets. And those granules are full of different proteins and really diverse proteins. And when platelets become activated, they release those granules and they release their protein payload. Because of this, they play roles in diseases such as malignancy, angiogenesis, inflammation, really almost any disease you can think of, platelets play a role in. However, I am not as focused on platelets as I am on how platelets are made. So platelets are made from we call their mother cell, the megakaryocyte. And while we've been studying platelets for a very long time, the megakaryocyte field is actually quite new. And I think part of the reason for this is because megakaryocytes are in the bone marrow and they're quite hard to access. So megakaryocytes develop from hematopoietic stem cells that reside mainly in the bone marrow niche, as you can see here. And hematopoietic stem cells are directed to differentiate along the myeloid lineage into megakaryocytes under the direction of thrombopoietin, or TPO. Just as EPO directs red blood cells, TPO directs megakaryocytes. Megakaryocytes are really, really unique cells, and I'll point out some of the reasons why here. So you can see labeled 2 is the nucleus, and the nucleus is actually polyploid. On average, megakaryocytes are about 16 N in the bone marrow, and that's because they undergo something called endomitosis, which is nuclear replication without cell division. They also have this highly invaginated membrane system, which you can see here, which is continuous with the plasma membrane and just has these folds throughout. And it's kind of a membrane reservoir, which is necessary for platelet production. So when megakaryocytes are mature and ready to make platelets, they sit up against the vascular sinusoid in the bone marrow and release these long fragments called proplatelets into flowing blood. And the turbulence of the blood makes the proplatelets undergo repeated rounds of fission events until they're finally the platelets that we know. And this happens really quickly. And eventually, like I said, the platelets are about 1 to 2 microns in size. So if we lay out this process horizontally, we have this very oversimplified here, showing hematopoietic stem cell differentiation into mature megs under the direction of TPO. I should note that this is really the only thing we know that directs this process. On the other side, our lab and others have done a lot of work looking into these terminal processes, how proplatelets are elongated, how they're released, and that's a microtubule-driven process. But what is completely unknown is what triggers round megakaryocytes to begin the process of proplatelet production. So this was first imaged by my mentor and now colleague, Dr. Joseph Italiano in 1998. And you can see that here, where we have a megakaryocyte converting all of its cytoplasm into proplatelet extensions. And this has been confirmed in vivo with intravital microscopy, where these proplatelets get directionally elongated into the flowing blood. Yet this is still a complete black box. We really don't know of any triggers that make this process begin. This kind of happens spontaneously in culture, although as scientists, we know there has to be some kind of trigger. And because of that, if you have thrombocytopenia, you're either given a platelet transfusion, or if you have chronic thrombocytopenia, you might be given a TPO mimetic, but that takes at least 5 to 7 days to begin to see increases in platelet counts, since that starts back at the hematopoietic stem cell. So it's really important for a variety of reasons and therapeutic options that we can figure out what triggers this. So we might be able to find therapeutic options to treat acute thrombocytopenia. So one of the observations that we had really early and has been known for really quite a long time is that inflammatory diseases can cause a reactive thrombocytosis or an acute increase in platelet counts. And whereas we know TPO can take days to do this, this can happen in a matter of hours in things like malignancy and autoimmune diseases. So because of that, it seems like this inflammatory induced thrombocytosis is TPO independent, which is really, really exciting. So this is kind of the basis for my lab. We use inflammation as a model of aberrant hematopoiesis. And in doing so, we hope to, one, figure out the role of platelets in the pathology of these diseases and reverse that, but also just kind of back engineer to understand some of the fundamentals of signaling pathways that could be triggering mature megakaryocytes to begin platelet production. All right, so that's a little bit of the background. As a postdoc, we had this exciting observation, or a finding I should say, that CCL5, or better known as RANTES, this chemokine was able to significantly and acutely, within hours, increase platelet production from megakaryocytes in culture, as you can see here. And data suggested that the reason for this was enhanced maturation. So these are the different N states of the nucleus. And what you can see here is normally in culture our megakaryocytes are 2N. However, when we treat with CCL5, on average, they're 16N. So they're undergoing additional rounds of endomitosis, enhanced maturity. And if we block CCL5 signaling through its receptor, CCR5, we can stop this from happening. Since we've actually done some, actually a lot of in vivo work, and we've seen that this is reproducible, and that CCL5 infusion can actually acutely increase platelet counts in normal healthy mice over 15%, starting at 4 hours, and so far it's been sustained for 48 hours. So this was really exciting, and something that no one's really seen. And this was in the absence of inflammation or increased white blood cells or increase in TPO. In fact, we see a bit of a decrease in TPO as seen with thrombocytosis. So with that being said, I began to think hard about how these inflammatory cues can actually access the bone marrow. So this is important because people have assumed it might be just a concentration gradient from the plasma or hand waved it. But to me, it seemed like there could be more of a direct messengers that could carry these inflammatory cues in different inflammatory states directly into the bone marrow. So one of the things that we see in a lot of these conditions is ongoing platelet activation. So platelets kind of have this like simmering level of platelet activation where they are expressing activation markers, but not yet degranulating their components. And when this happens, these platelets release extracellular vesicles into circulation. And I don't think I have to convince this audience that extracellular vesicles, or EVs, are a direct and efficient means of cell-to-cell communication. So I got really excited about this idea and went back and looked at some of the immunocytochemistry and immunohistochemistry we had done of femurs, looking if, in fact, there might be EVs of platelet origin in the bone marrow. So these are just other experiments. They were controls from other experiments. And what you can see here is if you look for CD41, so CD41 marks megakaryocyte lineage and also megakaryocyte progenitors. You can see that this is throughout the bone marrow here. And you can actually see quite a lot of punctate staining here for p-selectin, which is a marker that's on platelet granules, but upon platelet degranulation goes to the surface. So it's a marker of platelet activation. So from these, we've come up with these, a lot of questions. So I should say this is under review right now, but my first foray into EV research. So I'm excited to hear feedback. So the question is, do these EVs come from platelets? So the thing I haven't told you yet is that megakaryocytes also make EVs. So it can be quite difficult telling the difference between mega EVs and platelet EVs. Since platelets are derived from megakaryocytes, they have the same surface markers. Are they more present during inflammation, and what is the functional consequence of this? So just a little bit more about megakaryocyte versus platelet EVs. So it's been reported that basically CD41 positive EVs are about 80% of circulating EVs in blood. So it's really only recently that people have suggested that MEGs make EVs at all. And that was a paper published by Rob Flaumenhoft and Joe Italiano. And those are suggested to be passively released. So it doesn't seem to be a pathologic process. So in healthy individuals, we suggest that the majority of EVs in circulation are actually of megakaryocyte origin. However, upon platelet activation, so as I said, in different pathologic situations, platelets activate and release EVs into circulation. And these can be internalized and reprogram different cells within circulation, like macrophages and neutrophils. Same true with MEG-EVs, actually. So a bunch of studies have been published, first in vitro, and now recently a Blood Advances paper by Escobar and colleagues in Blood Advances showed in vivo that MEG-EVs can in fact reprogram hematopoietic stem cells within the bone marrow to become megakaryocytes. So in my mind, a lot of this work has looked at MEG-EVs within the bone marrow component and platelet EVs within flowing blood. But our unique angle of this is really showing that crosstalk, that platelet EVs could leave circulation and go into the bone marrow to carry their cargo. And one of the reasons why this is exciting and important is because platelets really act like sponges and take up things out of the plasma. So this is some data that I stole from Eric, so thank you to Eric. And he'll actually be giving this talk next week, so please tune in for that. But what this shows is that lupus mice, so this is a model of lupus mice where they age into the disease. So pre versus post-disease, their platelet transcriptome is very, very different. We've also done some proteomics showing the same. There are things that are upregulated and downregulated. Really, the purpose of this is just to say that platelets in disease states are quite different, and they can take things out of circulation. And we think that the platelet EVs could be acting as messengers, really relaying that pathologic plasma state straight into the bone marrow. So let's get to some data. Just to show you that I did my due diligence, we did characterization of these, partially because not a lot of work has been done on MEG-EVs. So we look at different sources, both murine and human. We have murine megakaryocytes from bone marrow and also derived from fetal liver cells, and human CD34 derived megakaryocytes. And that we just looked at the EVs per cell. Of course, they make a lot more than platelet derived ones. So these are platelets activated with thrombin and collagen, which is a physiological agonist, or LPS, which will match our in vivo assay, which I'll show you shortly. And and this fits with previous work. Platelets are quite small themselves, so it makes sense they don't make that many EVs. And we're basically selecting for an average size between 100 and 200 nanometers because we're only doing a 17,000 spin. We're not doing a really hard spin. So we're kind of enriching for those larger EVs and probably not looking at exosomes at all. We've also done electron microscopy and immunogold to confirm CD41 positivity and the number of CD41 positive sites per cell. So this is perhaps not that exciting, but a really important slide, and looking at the characterization of surface markers. So basically what we hope for is that we can kind of create a fingerprint to tell the difference between Meg EVs and platelet EVs. And then if we flushed marrow, we'd be able to say, okay, these are positive for this, so they have to be platelet EVs versus Meg EVs. Unfortunately, it was not that straightforward, but it was quite interesting. So basically we gated for the CD41 positive events, so it should be only from Megs or platelet EVs. And then we looked at positivity for two markers, CD62P or P-selectin, and Nexin 5 positivity, which binds to phosphatidylserine. And these are two things that have previously published to only be on platelet EVs and not at Meg EVs. So what you can see is that platelet EVs do seem to be enriched for this double positive population, P-selectin positive and phosphatidylserine positive. And Meg EVs do seem to be CD41 positive, but don't have what we would call these activation markers. So that's exciting and nice. However, it's not really strong enough for us to be able to definitively say on flushed marrow what is derived from platelets or megakaryocytes. So because of this, we have used this adoptive transfer model. So to do this, we use the c-MPL knockout mice. So these mice have very few megakaryocytes and have severe thrombocytopenia because they can't signal through TPO. So what we did is we added normal platelets to these mice to reconstitute platelet counts that are also fluorescent. So now we have two ways to track these platelets, both from fluorescence and MPL positivity. We then add LPS to activate platelets and therefore generate more EVs. And we can then quantify EVs by flow cytometry and immunohistochemistry. Okay, so first from our raw flow data, here's some example images. You can see when we add LPS, we have more of CD41 positive small particles that are smaller than our platelet gate here. And it's really nice that these mice have low platelet counts to begin with, because now our fluorescent cells are over 50% or over 60% in circulation. And even after LPS administration, they're about 40%. All right, so on to the exciting stuff. Here's our positive control. When we section the femurs, you can see these are just normal mice. You can see that the MPL antibody binds really nicely to Meg progenitors, maybe a few EVs here. Conversely, the negative control is quite negative. It's a surprisingly good and specific antibody. And what you can see here is that after we infuse platelets, we see this punctate staining really all throughout the bone marrow. Maybe even bound to a cell here. It's a bit overextension. And this is vastly increased when we add LPS. And we suggest that the reason that this is increased is because there are more EVs, not because that the EVs have more access to the bone marrow, but that's something we're still trying to parse apart. The caveat here is also that the procedure itself, I'm sure, triggered some inflammation. So I think this is our healthy and this is our no LPS, but they're not quite comparable. Undoubtedly, we see MPL positive events throughout the marrow, not just in the vasculature. And the only place where that is derived from was from the infused platelets. So we went on to quantify this, and we did something really just straightforward here, where we just flushed the entire marrow and looked at the percentage of all marrow cells that have fluorescence. So that suggests that are bound to EVs. And you can see here, over 8% were bound to EVs, which is actually quite extraordinary for all bone marrow cells. We also sorted for EV positive, CD41 positive events to see if they were bound to MEG or MEG progenitors. So these were sorted and then fixed and stained with just host. And you can see this MEG, nice big polyploid nucleus with EVs bound. So again, we see these penetrating into the bone marrow, binding to cells, and of those cells, some are CD41 positive cells. We haven't done an exhaustive characterization of what kind of cells they bind to. So we've also done this in an immune model with Eric Bouillard, but I'm not going to go into this. As to say, we don't think this is specific to a certain kind of inflammation. We think this is a more general process. So we've begun to also look at this in vitro, the binding to megakaryocytes. Again, here's our characterization and our immunogold of the PEVs that we're adding to the MEGs. So the MEGs aren't labeled at all, just the nucleus. So what you can see lighting up the membrane is the binding of the EVs to the cells. And it happens really fast within 30 minutes and sticks around for a while. We've begun to do more quantifying this, but a lot of it is visual, as you can imagine. All right, so is this important? Am I just studying a mouse phenomenon? Woke up in the middle of the night in a sweat thinking, okay, is this relevant in humans? And really lucky to have some collaborators in Moscow that were able to do some studies for us. So these are healthy patients, we'll say. They were having marrows done to confirm remission from ALL. So they're not exactly healthy, but they were all found to be in remission. And on the flip side, you can't really take marrows from healthy people. So this is a rather healthy cohort. And we were able to not only take marrow aspirates, but also autologous blood to try to account for any blood that leaked into the marrow and compare them head to head. So what you can see right off the bat is that just all EVs, the total number is significantly more in bone marrow than in peripheral blood. And then when we look specifically at the markers that should be MEG EVs and platelet EVs, those are also significantly more, even more so the ones that looked more like platelets with CD62 positive and PS positivity. We went on to look even more closely at this. You can see our gating here. So we looked at dual positivity. Well, here we looked specifically at the CD41 positive EVs, and then looked at the same markers we did in our characterization. And what you can see is, again, the PS positive are more enriched in the bone marrow. And these are more likely to be from platelets. And that should be the case in healthy patients. And conversely, in peripheral blood, we have more CD41 positive. So, I mean, this is by no means a definitive saying that there are only platelet EVs in the bone marrow, but it highly suggests it. And since we can't do pulse chase experiments in humans until we have better markers, this data, however, is very encouraging. So lastly, I want to end with this experiment that we were really hoping to do in vivo, but we've had to close our labs down, as I'm sure all of you have as well. But I'll show you this in vitro data that is enticing, which is really the most important thing. What is the functional outcome of this? So to do this, we took bone marrow from the Mipple knockout mice and flushed it and put it into culture and added TPO. And then in parallel experiments, we added EVs from wild type mice, so Mipple positive again, and fluorescent. So this corresponds to here. Even if you give this TPO, of course, nothing happens because they don't have the receptor. So these cells remain very small, as you can see throughout. However, when we add EVs to the culture after 3 days, we see a significant rescuing of megakaryopoiesis. So you see these big cells were able to form. And even more exciting is the fact that almost all of them were fluorescent. So this suggests that the ability to become megakaryocytes was restored to them from their interaction with EVs. And we're looking into that mechanism, of course. However, here it is quantified. So this is just looking at CD41 positivity. So it's also EV progenitors. So it perhaps underestimates the difference. However, there was an increase in CD41 positive cells when you added back PEVs in the presence or absence of TPO. So it might be a TPO independent mechanism, but we're not quite sure of that yet. In addition, you can see that the vast majority of these CD41 positive events were microparticle positive. In addition, this is just some raw data to show you that we weren't just staining for aggregates. It's hard when you're adding CD41 positive things and then looking for CD41 positive things. But the CD41 positive, PEV positive events were the biggest events. And to further confirm this, as we did in vivo, we sorted these by flow and spun them down. And again, you can see these megakaryocytes that are bound, or megakaryocyte progenitors being bound by EVs. So with that, I'm gonna end with this incredibly oversimplified model where we have inflammation, and our lab has historically studied CCL5. So that causes increases, which we think fed back an increased platelet count. But now we've added this kind of like fast track where the inflammation also causes platelet activation, releasing EVs. And we think those could be going directly into the bone marrow and binding to bone marrow cells, perhaps MEG progenitors, and delivering their cargo to direct them into becoming more megakaryocytes faster. So we suggest that this could be a new player in inflammation-regulated hematopoiesis, and in fact, it could be relevant to any disease with a thromboinflammatory component. All right. With that, I would like to thank all the members of my group. Shauna French and Kirill Butov did a lot of this work. And I have an amazing support system at Harvard and Brigham and Women's Hospital. Also, some of the work was done in the lab of Eric Bouillard, and we've done a lot of this in collaboration with him. So thanks to Eric for being a great collaborator. And I am really happy to take questions. I think we're going to do that at the end, but please save them up because I am looking forward to your comments and suggestions. So thank you everyone for tuning in.
Okay, thanks very much, Kelly. Laura, would you go ahead and start your screen share?
Is that good?
Yeah, you're good to go.
Good.
Okay. All right. Well, thank you so much. Thank you, Carolina, for all your work in setting this up. And thanks to all of you for sticking around to watch. I'm going to be telling you about our work in exploring the effects of aging on the total hematopoietic stem cell population in marrow and a potential role for bone marrow-derived EVs in modulating this aging phenotype. And I'm actually going to start just introducing you to some of the work we did in young murine marrow that was unseparated whole bone marrow stem cell capacity, just to explain why we started looking at the effects of aging on the total unseparated marrow population in the first place. And then I'll go through some of our data on the aging of hematopoietic stem cells in unseparated old marrow, and then again the role of bone marrow-derived EVs, our preliminary data. This is all unpublished work, so welcome suggestions and comments. So this is the hierarchical model of hematopoiesis. It really holds that there's a very rare, unique subset of long-term hematopoietic stem cells. At the top of the hierarchy, they have tremendous self-renewal potential, and they have pluripotency. And then through a series of relatively well-ordered differentiation steps through committed progenitors, they give rise to these more lineage-restricted peripheral blood cells. And this model holds that these LTHSCs, or long-term hematopoietic stem cells, are predominantly quiescent. And really virtually all the studies in the stem cell field in hematopoiesis have focused primarily on these purified stem cells, the quiescent ones. And there's multiple techniques to isolate hematopoietic stem cells. Two of the most common ones are using supravital dye techniques like dye exclusion and side population. And I think even more commonly immunophenotype profiling. And this is just an example of isolating hematopoietic stem cells through immunophenotype profiles. So I work with murine hematopoietic stem cells. So in this example, we flush whole bone marrow or crush bones and harvest the marrow. And then you incubate the marrow with antibodies directed against lineage-specific epitopes. And then any antibody-bound lineage-positive cell is discarded. That's about 96 to 98% of the marrow, so it's a huge chunk of the marrow. And the remaining 2 to 4% of the cells are lineage-negative cells. And it's from this population that the more purified, tremendously potent stem cells are isolated, and that's through both positive and negative selection for a variety of markers. I've just listed some of the more common ones, but there's a whole host of stem cell markers, positive and negative selection for stem cells and the multipotent progenitors. And using FACS to isolate these cells, you wind up with a very small purified population of stem cells. This is, again, just an example, lineage negative, c-KIT pos, SCA pos, and then the SLAM markers, CD150 positive, CD48 negative, and then murine CD34 negative. So a highly purified stem cell, again, tremendous self-renewal potential, limited or multipotent, and these are predominantly quiescent. Now, a lot of studies have shown very reproducible and strikingly reversible shifts in HSC phenotype linked to cell cycle in vitro. And that is to say, if you take purified stem cells and put them in culture in a cytokine-driven cell cycle culture system, you can synchronously walk them through cell cycle. And then at different time points in cell cycle, there are loss, gain, loss, and then regain of phenotypes such as homing and engraftment, differentiation, predilection, all linked to cell cycle state. And it was unclear if these were based on artifact because it was in vitro and a cell culture with cytokines, especially given that these highly purified stem cells are predominantly quiescent in vivo, as we know. And so we switched to looking at in vivo transplantation studies. And I will just sum up what we found. This has been published, but the bottom line, using BRDU in vivo labeling, propydium iodide, we used tritiated thymidine suicide and Herx and pyridine staining with transplant to essentially show that there is indeed a large population of actively cycling cells in whole bone marrow that seem to be lost with purification. And so here, this is the purified stem cells, a very small, rare population engrafting in G0. And what we found, and again, this is already published, a huge population of stem cells that are able to engraft not only in G0, G1, but also in SG2M, leading to the simple hypothesis that we're somewhere along the line losing these cycling stem cells during conventional purification techniques. And this fits with a concept where there's actually a broader population of cells in marrow, in whole bone marrow, that have stem cell capacity. They're going through cell cycle and thus changing their phenotype and functional potential with cell cycle transit. And these are the cells that are getting lost with conventional purification. And that data set is what triggered our work in the aging hematopoietic stem cell field to look at what's happening to the cycling population with aging. And the current concepts in HSC aging are that with aging, the number of hematopoietic stem cells actually increases, but their reconstitution potential decreases. And so their number goes up, their function goes down, and then they are myeloid biased. And whether that's because the hematopoietic stem cells are actually changing towards a myeloid bias during aging, or there's an expansion in myeloid biased clones during aging is unclear. But the end result is increased numbers, decreased function, a myeloid bias that is blamed for increased prevalence of myeloid diseases such as AML and MDS, and decreased immune responses. And again, I'd like to point out that these studies are all based on immunophenotypically defined HSCs. And so we set out really to look at what was happening with our cycling stem cell population in aging. And I just want to really quickly go through our transplant model because most of the slides are based on this experimental design. So we took donor bone marrow cells from CD45.2, C57 black 6 mice and combined them competitively with whole bone marrow from congenic B6 SJL CD45.1 mice. And then this mixture of cells we inject via tail vein injection into lethally irradiated host CD45.1 mice. And this irradiation is actually in 2 split doses, 3 hours apart. And then at various times post-transplant, typically 1, 3, and 6 months, we collect peripheral blood and do flow cytometry for donor and host CD45.1 cells or competitor cells. And so a lot of the graphs you'll see, there's percent donor chimerism on the y-axis, which is essentially CD45.2 divided by the total number of cells times 100, and then on the x is either time post-transplant or cell dose. So the first thing we did was to quantify the number of stem cells within old unseparated whole bone marrow. So this is in the absence of any purification as compared to young whole bone marrow. So again, we started with our 24 to 26 month old CD45.2 mouse, harvested total whole bone marrow, and then serially diluted it and mixed it with a constant number of young competitor whole bone marrow cells, injected them via tail vein into lethally irradiated young mice, and looked at peripheral blood chimerism. And we did the same with young in parallel as a control. And so this is all 6 months post-transplant. So again, on the y-axis is percent donor chimerism, and on the x here, again, this is all 6 months post-transplant, but donor cell input with decreasing cell dose as you go to the right, and a constant dose of competitor cell input. The purple line is the mice that received old donor, and the green line is the mice that received young donor. And as you can see, at every dose, old whole bone marrow outcompeted young. And actually by Poisson distribution using LCalc software, we estimated 1 in approximately 10,000 cells in marrow. In the old marrow were stem cells, whereas only 1 in 40,000 in the young were stem cells. And given the data that the old purified stem cells are myeloid skewed, we looked at lineage. So if you look at the left graph, this is percent donor derived peripheral blood. These are the mice that got old donor cells, and green is the mice that got young donor cells. And there is a minimal but almost no difference in the myeloid to lymphoid ratio between old donor and young donor. So it was statistically significantly different, although if you look at the ratio, it is within reference range, so about 20 to 30% myeloid versus 70 to 80% lymphoid. And on the right is the competitor-derived peripheral blood myeloid to lymphoid ratio, just as internal control. So this is the competitor young marrow in the mice that received old donors, and the competitor marrow in the mice that received young donors. So again, robust transplant activity and minimal myeloid skewing. And we tested in secondary transplant to test stem cell capacity. So these were mice that were initially transplanted with either old donor or young donor whole bone marrow with equal amounts of young competitor. And 6 months post-transplant, we euthanized the mice, harvested the whole bone marrow, and then injected into secondary fresh recipients. And again, percent donor chimerism on the y-axis, and then this is time post-secondary transplant. These 2 bars are the input donor chimerism. So you can see the old donor really outcompeted its young competitors in the primary transplant, and it maintained robust engraftment even in secondary transplant out to 6 months. And looking at the myeloid to lymphoid ratio, again, percent donor derived peripheral blood cells on the Y, and this is all 6 month post-transplant data. This is no significant difference in the myeloid to lymphoid ratios in the mice that received old donor versus young donor. And if this were old purified stem cells, there would be significant myeloid skewing here. So it was interesting. Now, because we saw a cycling population in our young marrow, we wanted to look at the cell cycle status of these cells in the old unseparated whole bone marrow. So we used tritiated thymidine suicide. We harvested whole bone marrow from either young or old mice. We incubated it either with just media, tritiated thymidine, which when you incubate with tritiated thymidine, it will incorporate into growing DNA strands during S phase and then kill that cell. So any cell transiting S phase during the incubation will be selectively killed. And then cold thymidine, unlabeled thymidine as a control. And we quenched the reaction after 30 minutes and had 3 groups that we then tested in competitive transplant. So again, any cell in this group, if it were cycling and also able to contribute to engraftment, it would be selectively killed in the tritiated thymidine group, and we should see a reduction in engraftment, which, not surprisingly, based on our prior experience with young marrow, we did see with the young whole bone marrow. So this is percent donor chimerism on the Y, time post-transplant on the X. And the green line is unmanipulated whole bone marrow. The blue line is unlabeled thymidine control. And you can see a significant reduction with the treated thymidine, suggesting the loss of cycling stem cells. And that's very similar to what we saw in the old marrow as well. This is unseparated marrow. Whole bone marrow alone, the blue is unlabeled thymidine control. And again, a significant reduction in engraftment with treated thymidine. And we repeated these studies using Hertz as well to show, again, there's a cycling population of stem cells in old marrow. And then this is just to show, so this is the data I just showed you. This is just to show that if you took the same young marrow and isolated a very purified population, the highly purified Lin-Neg, CKIT, CKIT pos, SCA pos, 150 pos, 48 and 41 negative cells, so a highly purified stem cell population, no effect with treated thymidine, which is exactly as you'd expect. These cells are quiescent and they're unfazed by incubation with treated thymidine. So again, these data suggest a cycling population even in old marrow. So to sum up here before I get to the vesicle data, the stem cell capacity of aged unseparated whole bone marrow does not appear to be declined with age as determined by limit dilution assays. Repopulation by old unseparated whole bone marrow shows minimal to no myeloid skewing in primary and secondary transplantation. And this is really in stark contrast to what has been defined for immunophenotypically purified old HSCs. There's a large population of cycling HSCs in young whole bone marrow. It seems to still be present in old whole bone marrow, but actually may decline with age. We do have some data, preliminary data that suggests that this population may not be as large. And so in conclusion for this part, there does seem to be a population of stem cells in unseparated marrow that has differential susceptibility to aging and to the aging microenvironment when compared to conventionally defined HSCs. And I say that because this data suggests that if you took an old whole bone marrow stem cell and put it into a lethally irradiated young host, you get robust engraftment with minimal to no myeloid skewing. And if you did the same with a purified old stem cell into a lethally irradiated host, that cell is, has poor engraftment capacity and myeloid skewing. So there must be something different. And so we felt like extracellular vesicles were an ideal candidate for modulating some of this differential susceptibility to aging phenotype. There is a lot of data, I don't have to remind this audience, but the data for EVs in aging and hematopoiesis is exploding. So EVs are implicated in the pathogenesis of numerous age-related diseases now. And this is just a very tiny tip of the iceberg smattering, but there have been a lot of publications showing that age-related changes in EV concentration and content occur. There's actually a really beautiful paper by Yoshida et al. Showing young murine plasma EVs injected into old mice increased lifespan. And Sishen Wen has some great data showing that young MSC-derived EVs can rescue radiation damage of HSCs. So not aging per se, but there are actually a lot of parallels to aging with the radiation damage model. There are decreased aging markers and mTOR pathway components in old MSCs that were exposed to young MSC-derived EVs, and it actually increased autophagy mRNAs in old Lin-Neg cells when they were exposed to young EVs. So there was a lot of data, but how EVs within the bone marrow niche are actually involved in aging of hematopoietic stem cells is not fully understood. So our goal was to start exploring this. And the first thing we did was to isolate EVs from young and old mice. And we did this by flushing whole bone marrow into PBS. Sorry, I keep doing that. To get young whole bone marrow and old whole bone marrow. And then we isolated EVs, and all of the experiments I'm going to show you are all just differential centrifugation is the only method so far that we've used. But this was 300G for 10 minutes, and then the supernate into 2000G for 30 minutes. And then we went right to the 100,000G spin. So we're collecting the total extracellular vesicle compartment. We washed it and respun it. And also, I will say that we jumped straight to functional analysis. So I will show you some nanoparticle tracking analysis. Unfortunately, with the shutdown, we don't have our EM and protein analysis characterizations yet. I will show you a little bit of microRNA analysis that we really have not delved into yet. And then again, the functional analysis. So I'll be calling these EV-enriched pellets, again, without the EM and the protein characterization. But this was our ultracentrifuge EV prep. So the nanocyte data, actually we didn't see any significant differences between the mean size of EVs from bone marrow derived young EV pellet or the old EV pellet. We did see a little bit of an increase in the number of EVs, so about a twofold increase in EVs from old bone marrow compared to young bone marrow. And then the first thing we did with these EVs is test just in vitro to see if they had any role in an aging phenotype. So this was done in collaboration with Jill Creeling at Brown University. And she isolated primary mouse tail fibroblasts and exposed the fibroblasts to either no EVs or 3 different doses of EVs in vitro, and then just determined senescence by senescence-associated beta-gal staining. So this is percent senescent cells on the y-axis, and then at different passages, these are MTFs that had no EV exposure, so just looking at the senescence through passage 2 through passage 5, and by passage 5, about 50% were senescent. But interestingly, when these same cells were incubated with 3 different concentrations of EVs, there was actually an increase in the percent senescent cells. So these yellow and gray bars here, statistically significant increase in senescence. So it looked like old EV incubation with these fibroblasts actually accelerated their senescence. So by passage 5, about 65% of the cells entered senescence versus the 49%. And we did the flip side with the young. So here again, this is percent senescent cells on the Y. These are the same MTFs with no EVs, and then incubated with EVs derived from young bone marrow and actually delayed senescence, statistically significant, mild, but significant decrease in the number of cells that entered senescence. So again, and consistent with a lot of literature out there showing that old EVs play a role in senescence. And so then we jumped to looking at the HSC phenotype. So we went to our transplant model, and I will show you our negative data first. These are young mice, so 6 to 8 week old CD45.2 mice. We either injected them with PBS via tail vein, the old whole bone marrow derived EV enriched pellet, or the young whole bone marrow derived EV enriched pellet. And we did this via tail vein injection, and we did it daily for 3 days, 2 times 10 to the 9th EVs per injection. And then we just let the mice sit for 4 weeks. So 1 month post EV injection, we isolated whole bone marrow, or the SLAM cells, what I'm calling here, which is LINNEG, c-KIT pos, SCA1 pos, CD150 pos cells. So these are cells from young mice that were exposed to either PBS, old EVs, or young EVs. And the question here is, can we accelerate an aging phenotype by exposing the marrow to old EVs? And then we mixed them with young, fresh competitors and injected them into lethally irradiated young mice and looked at peripheral blood chimerism. So this is the data from the whole bone marrow from young mice. And as you can see, this is percent donor chimerism again on the Y, 1, 3, and 6 month post-transplant. And then the blue is whole bone marrow control, purple is whole bone marrow plus old EVs, and green is whole bone marrow plus young EVs. And there's absolutely no difference. So exposure of the young mice to old EVs didn't change engraftment capacity. And we saw the same with the SLAM. So we get about 60% engraftment, and it's unchanged whether these SLAM cells were from mice that were exposed to old EVs, young EVs, or PBS. Which actually makes some sense in retrospect, which is why I showed you our old whole bone marrow data. That old whole bone marrow actually engrafts better than young marrow. And so it is not so surprising in retrospect that old EVs from bulk old whole marrow does not alter this engraftment capacity. And then we looked at lineage again. SLAM cells, as they age, get more myeloid skewed. This is young whole bone marrow, looking at the percent of donor-derived peripheral blood cells that are myeloid versus lymphoid from the mice that saw PBS, the mice that saw old EVs in purple, or the mice that saw young EVs in green. And again, there's no change in the skewing, which in light of our data with whole bone marrow, unseparated old whole bone marrow showing no myeloid skewing is not so surprising. And the same with the young. We saw no myeloid skewing and no change with the EVs. So here's the reverse. This is old mice now, seeing if we can rejuvenate with young EVs. So old mice, 24 to 26 month old, injected with PBS, old whole bone marrow derived EVs, or young whole bone marrow derived EVs. And these again are the EV enriched pellet from the ultracentrifuge spin. And the same thing, 2 times 10 to the 9th EVs IV daily for 3 days via tail vein injection. 1 month post IV injection, we isolated whole bone marrow, old SLAM cells, and then looked at peripheral blood chimerism. So here's the data. This is sort of an interesting and somewhat surprising result. So percent donor chimerism on the Y, time post-transplant. And again, blue is whole bone marrow from the PBS exposed controls. Purple is the whole bone marrow from mice that had seen old EVs, and green is the old whole bone marrow from mice that had seen young EVs. And actually, there was a statistically significant drop in engraftment capacity by the old marrow that had been exposed to young EVs, which actually mirrors our old and young whole bone marrow engraftment studies in the absence of EVs. We saw the same thing with SLAM. So this is, blue line is SLAM cells that were from old mice that saw PBS. And as you can see, I don't know if you remember from the other graph, but already you can see that they're not great at engrafting. So the young SLAM cells at the same dose were about 60% engraftment, and this is about 20% engraftment. So their functions declined as expected. And it seems to be decreased again with young EV exposure, and this was not statistically significant, just a trend. And there's huge variability in the purified HSCs from old EV exposed mice. And really interestingly, when we looked, we isolated these by flow, so we had counts, and actually the SLAM cells from the mice, the old mice that received young EVs, just 3 doses a month prior, had 3,000 SLAM cells per mouse, versus 18,000 SLAM cells in the old mice exposed to PBS. So very interesting, and again, fits with the known increase in immunophenotypically defined stem cells with aging. So it may be that the young EVs are able to control the population size, but this is just, it's preliminary data, so we've got some work to do. And then this is just summarizing that data. So again, percent donor chimerism. This is old whole bone marrow from mice that were exposed to young EVs, a significant drop. And this is old SLAM. And again, maybe not statistically significant, but maybe a trend towards a decrease in engraftment with the young EVs. And then really interestingly, here is the donor-derived peripheral blood cells. So if you just focus on the left panel, this is the mice that received old whole bone marrow. Either from mice exposed to PBS, old EVs, or young EVs. And as you can see, by and large, there's not a huge amount of myeloid skewing in any of them, and there's no statistically significant changes. But very interestingly, and it may be something, it may be nothing, but we definitely have to repeat these studies. These are aged SLAM cells from mice that received PBS, old EVs, or young EVs. This is the standard myeloid skewing that is classic for old SLAM cells. So instead of a 30-70, this one is about a 64-36%. And there does seem to be a slight reversal, a huge variability, but a slight reversal in this myeloid skewing trend when the mice were exposed just to 3 doses of young EVs a month prior. So intriguing, but definitely have some follow-up studies that we need to do. And then again, it'll be very minimal microRNA data. So this is just, we took old EV enriched pellets from bone marrow and then the young EVs and looked at some microRNAs. But we did see some hits that are interesting and worthy of follow-up. So these are down in old EVs, and these microRNAs were up in old EVs. And we have a lot of digging to do, but the ones that caught our eyes, MIR29A, MIR105, MIR24, 21, these are all have clear roles in hematopoiesis, senescence, and inflammation. So it'll be really interesting to follow these up. And then just in the last minute, I just want to talk about some old data that has been published, but I think is relevant to this project. And that is that as cycling cells transit cell cycle, they change phenotype. And the question is, does the cell cycle phase affect extracellular vesicle communication? And these are studies that were published by Jason Aliota in Stem Cells in Development. And he used a very, very well studied in vitro model where extracellular vesicles from lung co-cultured with marrow cells actually induce the marrow cells to produce lung-specific epithelial markers. So you can have a very nice readout of whether or not the marrow cells were affected by the extracellular vesicles. And he either cultured lung across a cell impermeable membrane, lung-derived conditioned media without the membrane, just with whole bone marrow, or lung-derived extracellular vesicles from lung. Co-culture for 7 days, collect the whole bone marrow, look at RNA, and then do RT-PCR for these 6 pulmonary epithelial markers and compare them to whole bone marrow cultured alone. And any whole bone marrow cell that has the presence of one of these has been influenced by the extracellular vesicle. And so he just overlaid cell cycle, and this is the last data slide I have. Actually, the methods in a data slide. So these are Lin, Neg, Scott, Paz cells. He synchronized them in cell cycle and culture using cytokines. And then at 0 hours, it corresponded to G0, G1. At 48 hours, corresponds to late S and early G2. And then overlaid them, depending on the time. He pulled them out either at 0 hours or 48 hours, overlaid them with vesicles, and then again looked at RNA 7 days later. And here's what he found. So this is fold difference versus uncultured cells. And these are the 6 pulmonary epithelial mRNAs he looked at. And you can see every single one of them, there was an increase when the cells were at G0, G1 at the time of initial vesicle exposure. So clearly there's some sort of cell cycle phase influence on EV communication, which I think is interesting in the context of our stem cell data. And so with that, just conclusions, there seems to be a population of stem cells in unseparatable bone marrow that has differential susceptibility to aging and the environment in the aging milieu when compared to conventionally defined HSCs. And bone marrow-derived EVs, based on this preliminary data, may alter the engraftment capacity within old whole bone marrow, specifically the old purified stem cells. They modify lineage commitment of old LSK SLAM cells potentially, and it looks like they may even modulate the population size. And the cell cycle phase may contribute to differential EV effects in the subpopulations during aging. And so obviously the niche is very complicated. A lot of us are focused on MSC EVs, but I think obviously there's a huge population of extracellular vesicles that are likely playing a role in stem cell fate modulation. And if there truly is a cycling population of HSCs in addition to this very quiescent purified population, it has intriguing implications for vesicle mediated communication that may be different than what we've been seeing with just purified stem cells. And so again, I think these data raise more questions than answered, and hopefully we'll get back to lab soon, but looking at the different subpopulations of EVs, and again, we looked at bulk EVs, not subfractionated based on size, looking at the differential susceptibility of different HSC populations, obviously characterizing the cargo and hopefully getting some insight based on the cargo into the mechanisms that may be modulating the population size, repopulation capacity, and lineage fate. And with that, I will just acknowledge Peter Quesenberry has been a fabulous mentor and now colleague and collaborator. Mark Dooner, who did a lot of the flow and has been instrumental in all these experiments, and the Cobri stem cell and aging flow cytometry core. And then a special call out to Sishan Wen and Jill Creeling at Brown, who were helpful with the aging experiments. And thank you for sticking around and listening.
Thank you so much, Kelly and Laura, for your amazing talk. It has been very interesting, and I'm sure that everybody has enjoyed. We have a lot of questions. We're going to start with question for Kelly. Please turn on your cameras before starting asking your question. Navneet, please go ahead.
Yeah, question to Kelly. Kelly, very nice talk. So question about platelet contamination or platelet presence. And what we saw is smaller vesicles, less than 100 nanometers, peaking at about 60. They did not have any platelet contamination. And larger vesicles did have. Have you seen something like this? How does your flow show that? Is it biased toward larger vesicles? Thanks.
We're definitely biasing just by our spin speed. We're biasing towards larger vesicles. As a method of simplification, we have looked at more like exosome-like things, and I would agree that they're not as CD41 positive, but it's quite complicated with platelets because they're small themselves, and we don't know what exosomes would even come from, if they're like whole granules that are released or things like that. So it's not as straightforward as with other cells. So, since it was kind of our first foray, we did bias ourselves towards what we think are more extracellular vesicles that are an active release product of platelet activation.
Thank you, Kelly. Diane, you're next.
Hi, Kelly. Thank you. Great presentation. I was wondering, are these EVs filament negative?
Yeah, so we haven't looked. We actually went back and looked at LAMP1, and it does look like LAMP1 can distinguish between platelet and megakaryocyte EVs on Western blot. However, we were just looking for surface markers, so we were kind of hoping that we could sort them by flow. So we have not done extensive characterization of differential protein expression and things. I think we'll probably have to go back and do a larger panel now. So we haven't looked at filament, but since people have been suggesting things, and we have a pretty long list of things to look at. So I think it's a really good idea. Yeah. And perhaps we could do immunocytochemistry or immunohistochemistry without the adaptive transfer once we have markers to tell them apart.
Yeah, and I was wondering, since these EVs are PS positive, do you know if they're from procoagulant platelets or from apoptotic platelets, and how are they protected from clearance to PS?
Yeah, it's a really great question. So I think we're looking to continue our collaboration with the lab in Russia. Mikhail Panteleev is an expert on procoagulant platelets. And I would say that, if I had to guess, probably the PS positive ones come from procoagulant platelets. And the other ones may come from not procoagulant platelets. I am not an expert in this, and I know this is a really hot topic for coagulation, but I've also talked with him, and he thinks this is likely as well. So we've thought about using microscopy to look at, the procoagulant or ballooning platelets and see what kind of EVs those release versus the other ones. And I do think they're actually cleared quite quickly from circulation. And I think that's one of the things that's really interesting about this, is while they're cleared very fast from circulation, they remain in the bone marrow we've seen them for days. So since they're not being, going through the spleen and being taken up by those cells, they're kind of protected in this niche. So I think that's one of the interesting points about them residing in the bone marrow.
Very interesting. Thank you.
Thank you, Diane.
Madeleine, do you want to go next?
Yeah. Hi. Thank you so much, Kelly, for your question, for your presentation. My question is, so you said you did some protein profiling. I think you used flow for that. I was wondering how you did that. Was it single vesicle or was it bead-based? Because I've tried bead-based protein profiling and I've run into a huge problem, and I'm looking for ways to troubleshoot that. Do you mind sharing how you did it?
Yeah, so it was by flow cytometry, but these were only surface markers.
Okay.
So, and we didn't quantify the amount of protein. It was only the percent of of cells that were positive for.
Okay, so yeah.
So it doesn't reflect the amount of that receptor on the surface of the EV, just whether the EV has it or not. The way we did quantification of the amount of receptor was by immunogold, so it's quite tedious.
Okay.
Yeah.
Do you have a paper where is any of that data published? Maybe I can look at it. Perhaps it would help me.
It's not. Hopefully it will be soon. But
Okay.
You can email me. I'm happy to share methods with you.
Okay, I'll do that. Thank you so much.
Yeah, of course.
Okay. And last question for Kelly, it's Kalyani.
Yes. So hi, Kelly.
Hi.
It was you had a great talk, and I think you're certainly adding to the traditional assumptions about hematopoiesis. So from your experiments, it is evident that the platelet-derived extracellular vesicles do bind to megakaryocytes. So I was wondering, since the HSCs and megakaryocytes share certain similarities, such as the shared dependence on TPO for maintenance and expansion, and they also share some cell surface receptors. So do you think that these platelet-derived EVs can also bind to HSCs and may bring about certain changes in the lineage commitment?
Yeah, that's exactly what I think. I think that we've been able to show it somewhat convincingly in vitro. Just as we were ready to, we had enough mice to put the EVs into the in vivo, this happened. But the thing we want to do most is basically do that experiment in the MIPL knockout mice and see if we can raise platelet counts just by the EV infusion. So that would be looking at megakaryocyte, but in addition, we'll flush the marrow and look at all the, you know, lineage and all the myeloid cells and see, because, you know, we're just looking at megs because that's what we're interested in, but there's no reason to believe they're not having a broader impact. So we really don't have a handle for that yet, but thank you for that question.
Okay, and yeah, and one more thing was, again, it is related to my first question, is that now with TPO, there are other interleukins that are also involved. So do you think that after the binding of the platelet-derived extracellular vesicles, do you think that there would be an upregulation in TPO as well as the other cytokines that are involved?
Yeah, so I'm thinking that this is probably not TPO dependent because it's very acute process. So it happens in our model in just a couple days, or in the other model in under 24 hours. And TPO can take a really long time. So it's a bit hard to parse apart because we haven't done a system with completely no TPO, which I think is something we could look at. But I think the more exciting outcome would be that they're delivering either, like you said, some other kind of chemokine or even some kind of like microRNA or something that's able to bypass the TPO signaling.
Okay, got it. Thank you so much.
Thank you.
Thanks, Kelly, and thanks for all the questions. We have one question from Andrew for Laura. Andrew?
Hi, Laura. Thank you very much for the talk. It was really good. So I had a question about whether you'd done any localization work to look for HSE localization differences between your old and young subsets.
We have not yet, but that's a great question, especially with relation to quiescence and whether they're on the endosteal surface. But yeah, no, we haven't looked at localization yet, but we would love to. We have looked at, so Sachin Wen actually did some studies looking at homing of vesicles. So when you inject the vesicles into a radiated mouse, they do seem to preferentially home to spleen and marrow, which is consistent with the notion that they tend to go towards damaged tissue. But we have not done homing with the stem cells yet. It's a great idea.
Okay, thank you.
Thanks.
Thank you so much. Thanks all the presenters and everyone for attending, and looking forward to see everyone next time.
Yeah, thanks very much, everyone. So this lecture is recorded, but not going to be deposited in WebEV Talk, but it's going to be online on YouTube for 24 hours from Kellie's channel, I think. Yeah.
Yeah, I'll share it with everyone.
Yeah. All right. Well, nice meeting you all. I hope to see you again next week. And in the meanwhile, please stay safe. Bye-bye.
Bye. Thank you, everybody.
Can we have sort of like a debrief session? Everybody has time. Oh, I wonder if Kellie already awake. Okay, I'm just going to open a new channel. Okay. Sorry.
Okay.